Hello class, this is your introduction into cell biology. You can find this Prezi linked from my website. It is called Intro and Endomembrane. To start, just with a couple of people, just so you can see their faces. Anton van Leeuwenhoek. He is the dead white guy who is the first recorded person to be able to look at what we think of as cells under a high-powered microscope. Followed by Robert Hooke, who coined the term cells in his observations of these cells within cork, because it reminded him of a prison. And that gets us back to cell theory. All living things are made up of cells. Viruses are not going to be classified as living because they don't satisfy this criteria. They are not made of cells. The cell is the basic unit of life, so a single cell contains all of the characteristics and abilities of any living thing. And finally, all cells come from pre-existing cells. They cannot spontaneously appear. Cells cannot be produced. Cells will only be the product of reproduction or the division of an already existing cell. Now, to look next at the idea of size, cells exist in this range between 10 micrometers and 1 millimeter. That's highlighted in yellow here in this figure. You can see down atoms and small molecules at the bottom, larger organisms at the top, and every one of these lines represents a power of 10. Now you can see that in the macroscopic world, in terms of the world that we can see with our naked eye, there are a few cells that we can see, mostly eggs, frog egg, chicken egg. There are cells that are very large in length, but not necessarily very wide, so they aren't as easy to see like some nerve and muscle cells. But most cells we need a microscope to be able to see, so we call cells microscopic. And most eukaryotic cells will be towards the top of this range, and most prokaryotic cells will be towards the bottom of this range. The very smallest cells down the very, very tiny bacteria, down the next power of 10, followed by viruses, some organelles, and some other molecules. Those things can only be seen with the use of an electron microscope, which, as opposed to a light microscope, which simply bends light using a curved piece of glass, an electron microscope will actually shoot electrons at the object, and depending on how those electrons bounce off, an image can be created with the use of a computer. Now, there's a reason why we have cells within the body, and that reason is very much linked to the fact that if we were just one giant blob and not made up of individual cells, we wouldn't really be able to move the materials into and out of the cell that we need to. So a couple constraints on cell size. If the cell is too small, you can't fit everything inside that's necessary. You can't fit in all of the machinery that's necessary to process energy, to make proteins, and do all kinds of other kinds of interesting things. If the cell is too big, then we can't get things in and out very efficiently. And if you look at this chart here, if we compare these three types, if the cell is very small, it has a large surface area, a small volume, and so it has a very large surface to volume ratio. The box in the middle has a large surface area, but it has an extremely large volume. And so the surface to volume ratio is much smaller. So there's a lot of stuff on the inside, but there's not as much room available to be able to get new stuff in and get old stuff out, namely getting things like oxygen and water and food in, and getting wastes out. So the body instead subdivides, and we have this little Rubik's Cube here, where we have a very large surface area because if we calculate each box has that surface area of 6 multiplied by the total number of boxes, and so the total volume is still pretty large. It's a large organism, but there's way more surface area, and so it's easier and quicker for things to get in and things to get out. Prokaryotic cells are going to be simpler, no membrane-bound organelles. They're small, and there's way more of them in the world than any other type of cell. You certainly need to know some of the particular structures that are in the prokaryotic cell, the flagellum, the capsule, the plasma membrane. Now, some of the things that are unique, the fact that the DNA is not concentrated into a nucleus, but it is free within the body of the prokaryotic cell. We see some animal cells do have flagellum, so that's not necessarily unique. The idea of a capsule, only some bacteria have this, and this is a coating that is on the outside of the cell wall. Many eukaryotic cells do have a cell wall, but the capsule is something that is unique. Moving forward, we have eukaryotic cells. Eukaryotic cells are a lot more complex because they have extra parts. Those parts are often bound by membranes, and that really helps to increase the ability of the cell to do much more specialized things. I will start with plant-like cells. Those are photoautotrophic things that would include both protists and plants. Now, within the plant cell, all of the highlighted parts are the things that you need to know. There's a lot of overlap between the animal and plant cells, but for any of these highlighted parts, you need to use your other resources to be able to determine what is certainly the name, and to match with that name what it looks like, what its structure is, and what its function is. So, for example, I could look at the chloroplast. We studied chloroplast previously with photosynthesis. The chloroplast is a membrane-bound organelle. It's actually double membrane-bound because there is an interior membrane within the chloroplast. And its function is to carry out the process of photosynthesis, which helps to capture energy from the sun to be able to produce carbohydrates. Now, animal-like plants, these are things that are chemoheterotrophic. So these types of cells are things that are obtaining food from outside of themselves. Like with some prokaryotic cells, there are many that have a flagella. And similarly, you need to be able to know what these highlighted parts, what their names, be able to identify them, what is their structure and what is their function. So you look at the lysosome. The lysosome is a membrane-bound organelle. Its function is to be able to digest toxins and worn-out cell parts to be removed from the cell. So similarly, study the rest of those and take a look back. Two other points about membrane-bound organelles. First is that they're very useful. They allow eukaryotic cells to specialize, and because of the fact that these organelles will provide these kind of separated, protected areas that can have a different concentration of enzymes, can have a different pH, they have lots of surface area for lots of reactions to be able to happen, and this is what allows different types of cells to be able to do different tasks within the body. So a heart cell, for example, is packed full of mitochondria because heart cells need to be able to process a lot of energy and do a lot of work continuously, day in, day out, throughout the course of an organism's life. So the fact that it has more of the organelles that it needs to do its job sets it apart from something like a nerve cell, which is going to have many fewer mitochondria because it doesn't have those same needs. You see within the pictures here all of the different compartments that are there within those cells. Those provide lots of different areas within that cell for different things to be able to happen. Now, the fact that these organelles exist also carry out many of the functions for life within the cell. So processing matter, that's going to happen. It's going to be a combination of the different membrane-bound structures within the cell, things like the vesicles and the ER that are transporting things from one part of the cell to the other. Those are serving to process matter. Mitochondria is going to be processed. There as well. Energy needs to be processed. So the mitochondria have a role in that. Chloroplasts have a role in that in plant-like cells. And then finally, process information. That information is going to be, that we're mostly talking about is DNA and RNA. So there is DNA that's contained within the nucleus. That information is going to be passed from the nucleus in the form of RNA out to ribosomes, where it's going to be turned into proteins. Some cells need to reproduce, so they have parts necessary for them to be able to divide. And then communication. So many cells have ways of being able to generate chemicals that will be able to be expelled from the cell and send a signal to either surrounding cells or other parts of the body in large complex organisms. Now finally, for this particular video, proteins. Now proteins are where most of the action happens. Here's a brief list of things that proteins will do. We'll cover this more in depth in a future unit. But generally speaking, they are the most important type of molecule by far within the body because of all the jobs, all the functions that they carry out. Now, the way that proteins are going to be built is using information that is contained within DNA. Each gene within the DNA contains information about how to build a particular protein. Now, within the nucleus contains all of this DNA. And DNA is an extremely long molecule. We'll study this a little later on. And so that DNA needs to be kind of carefully coiled and wound up within the nucleus to be able to fit. Because it's several meters worth. If you took a single strand of DNA out of the nucleus and stretched it out, it'd be a couple meters long. And that's pretty easy to tangle if you've ever dealt with something with a very long piece of string. And so there's a lot of very careful coiling and wrapping that goes on to make sure that that DNA doesn't make a mess. So DNA is going to be stored here, it's going to be copied here, and those copies can be sent out to other parts of the cell. And then additionally, ribosomes. Ribosomes are the place where information from the DNA is going to be sent, and there's two subunits, two parts to the ribosome, and its job is to be able to make proteins. So this is where that action happens, where protein synthesis, using that RNA that came from the DNA, will make a particular protein. And those proteins then, because many times this happens on the rough endoplasmic reticulum, as we'll see later, those proteins can be packaged and sent out. And that's what we'll cover in our next video as we look more at the different relationships between those membrane-bound organelles and how they work to be able to transport materials into and out of the cell.